Good evening, everyone, and welcome to our series of talks entitled Reasons to Believe. Throughout this series, we'll be looking at the reasons why we as Christians have the faith that we do. Why do we believe in God? Why do we believe in His Word, the Bible? Why do we follow His Son, Jesus? And what does that mean for us as Christians? Tonight, we'll be looking in particular at the origin of life. These talks will be short, only 15-20 minutes, just to provide an overview of each topic. If you are a Christian already, we hope that you might find these talks encouraging in your walk towards God. If not, we hope that you might encounter some new ideas and be inspired to look more closely. A little note then on God and science. Sir Isaac Newton, one of the greatest physicists who ever lived, published his magnum opus, the Principia Mathematica, in 1687. This great work contained the summation of Newton's theories on the laws of motion and universal gravitation, and it's still considered today to be one of the most important single contributions to science ever produced. Attached to this work was an essay, kind of appendix, in which Newton outlined various philosophical corollaries of his scientific study. And in this essay, Newton makes the following statement. This most beautiful system of the sun, planets and comets could only proceed from the counsel and dominion of an intelligent and powerful being. In a nutshell, Sir Isaac Newton believed in God, an intelligent, powerful being. We find that this belief in God was not limited to Newton alone. Johannes Kepler, the famous astronomer whose work preceded Newton's by about 70 years, famously exclaimed, "O Almighty God, I am thinking your thoughts after you." Gregor Mendel, the so-called father of modern genetics, was an Augustinian friar. He is the one whose study of heredity in pea plants, which he concluded in 1863, is still taught in schools today. For these scientists, their belief in God was not at odds with their scientific study. In fact, their belief in a creator inspired them to look for order in the natural world. As one modern commentator has put it, the fathers of modern science, Copernicus, Galileo, Kepler, Newton, Boyle, and many others, saw design in the universe and indeed were inspired to discover the laws of nature because of their belief in a transcendent lawgiver. Belief in God, then, was entirely compatible with scientific inquiry for these individuals. The two went hand in hand, a belief in the order of the universe spurring theory and experimentation to delineate and quantify that order, and with the results of these inquiries serving to increase awe and wonder at the power of the Creator himself. However, when Charles Darwin published The Origin of Species in 1859, his theories added to a growing consensus that the entire world could be explained in material terms. There was no need to make reference to a supernatural creator. Every observable thing had an observable cause. The theory put forward by Darwin goes roughly like this. You're probably familiar with it. Given a population of living organisms, when one organism undergoes a random change, it will be altered in some way. This change is called a mutation. If this mutation proves advantageous, the organism is more likely to survive and reproduce. If not, then it is more likely to die without offspring. In this way, over successive generations, advantageous changes, mutations, will be preserved, and the species will have evolved. Occasionally, these mutations might add complexity or functional parts, which would explain in purely natural terms the dizzying array of capabilities on display in the natural world. Each part of each organism is the result of many, many random mutations, and the fact that they all appear so wonderfully well suited for their purpose is simply because the best survived and the rest died out. Whether you believe that or not, tracking back in time, the logical corollary of this is that all complex life, although it appears designed and purposeful in its current form, all complex life can be traced back to simple life. And although Darwin did not deal with this question in detail, it was not then unreasonable to suppose that simple life could have arisen spontaneously from non-living things. Thus, the apparently sticky problem of the appearance of design in living organisms was solved, with reference only to natural causes. God was not necessarily dead, but he just wasn't needed anymore. Now, when Darwin was writing in 1859, this might have been plausible. To the Victorian observer, life in its lowest form was simple. Microscopes of the time saw single cells, but these homogeneous, structureless, pulsating blobs of what scientists of the time referred to as protoplasm were fundamentally unremarkable. Since then, however, science has made great strides in understanding what life is actually made of. Throughout the 20th century, scientists came to understand that the cell actually performs a vast array of complex chemical functions. And nowadays we know that the cell is a hugely complex and well-ordered machine. Take the cell membrane, for example. When this part of the cell was hypothesized in the late 19th century, it was presumed to be a simple film of lipids or fats, which served as a sort of basic barrier between the contents of the cell and the unpredictable exterior environment. Nowadays, it is understood to be a highly specified and complex system of not only a lipid-based chemical barrier, as originally supposed, but also a system of molecular gates, which can open and close to allow different compounds in and out as the cell requires. Rather than being a simple balloon-like membrane, the cell membrane is much more like passport control at Heathrow Airport, fully staffed and functioning, if you can imagine that. And if this subject interests you, I'd suggest take half an hour, maybe now or sometime after this talk. Find a diagram of the cell, which lists out the organelles or the parts of the cell, the membrane, the nucleus, mitochondria, and so on. Choose one of them and look into it in more detail. And I can guarantee there is nothing in there that will fail to astound you. Every single part of the cell must exist and work before the cell functions as a whole. And the cell is the simplest form of life, a dizzyingly complex and impeccably engineered molecular machine. Surely it is impossible not to see a creative hand at work in this. Now, aside from the mechanical complexity of the cell, there is one other aspect of this subject, of the origin of life, which I do want to touch on. And that is of the information content of living beings. And at this point, I'll take the opportunity to recommend a book by Stephen Meyer, Signature in the Cell, which argues strongly for design specifically based on the information content of DNA and other biological systems. There are a number of excellent books on related subjects, but that one is one I've enjoyed recently, and he deals with this subject very thoroughly. So the information content of the cell. What is information? Well, there are a few ways to think about it. On the one hand, information is knowledge. If you tell me something I didn't know before, you have given me information. Perhaps you told me a friend's phone number, or gave me instructions on how to put up a shelf. In both cases, I have gained something from the exchange, something which is not physical, but which leads to physical effects. The something is information. And there is another aspect of information, particularly apt to this question of the origin of life. And this is the way in which different sequences or arrangements of something will produce different effects. The easiest example for our purposes is language, written information or text. For example, a book, as published, will read in a certain way and convey a certain message. However, if you rearrange all of the letters, the message will change. If you rearrange them at random, there will likely be no message at all. Likewise, instructions in the form of computer code. Any programmers listening will be familiar with the catastrophic impact of missing even a single comma or misspelling a single prompt. Incredibly, exactly the same principles apply in the living cells which we are all made of. DNA is the most famous example of this. Think of that now famous double helix model of DNA, which encloses a very specific sequence of 4 bases, which we give the letters A, C, G, and T. This is your genetic alphabet. And just as with the words in a book or the instructions given to a computer, the letters must be arranged in exactly the right order in order to produce the desired results. Precisely how the results are produced is still the result of subject of much scientific inquiry, but the fact remains that the bases, the letters of your genetic alphabet, must be in exactly the right order, or the cell will not function and reproduce as it needs to do. Now, as it happens, exactly the same thing is true of proteins, another component of molecular life. Unlike with DNA, which has an alphabet of 4 letters, proteins are constructed from an alphabet of 20 amino acids. And these join together in a linear sequence, just like words on a page. Depending on the precise arrangement of these amino acids, the protein chain will fold into a very specific shape to perform functions within the cell. And the misplacement of a single amino acid in the chain will mean that the protein does not fold correctly and becomes simply an expensive chemical mess clogging up the cell. Now, why is this helpful in demonstrating that the cell must have been created? It cannot have arisen spontaneously. It's useful because the information content allows us to calculate precisely the probability that it arose by chance. So you will appreciate that just as there are many ways to arrange the letters in a book, there are also many ways to arrange the base pairs in DNA, or the amino acids in a protein. For example, with DNA, I have 4 possibilities for the first base. I just need to choose one of the bases A, C, G, or T. For the next base, I also have 4 choices, regardless of what I chose for the first one. Therefore, when choosing the first 2 bases in DNA together, I have 16 choices, 4 times 4. Every time I increase the length of the sequence, I multiply the number of possibilities by the number of possible characters. So for a 3-base sequence, there are 64 possibilities. For 4 bases, 256. For 5, 1,024. Very quickly, the number of possible sequences becomes staggeringly large, and with that, the probability of choosing the correct sequence becomes staggeringly small. To cut a long and numbers-heavy story short, a very, very conservative estimate which allows every single atom of the universe, every single unit of quantum time, to rearrange the letters of DNA. The probability here of producing a single, minimally complex cell is around 1 in 10 to the power 40,000. Not 1 in 40,000. That's still very unlikely, but we could perhaps suspend our disbelief for a moment. No. The chance is 1 in 1 with 40,000 zeros. If I were to say that number like this, 1 in a million, 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 saying million twice every second, I'd be reading that number out for almost an hour. And if you still suspect that life might have arisen by chance, try reading that number out, a million, 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 and see how you feel an hour later. So what are we to conclude? I would contend that the message of the Bible is not just infinitely more hopeful and joyful in this troubled world, but also far more intellectually satisfying than the alternative. As David writes in his 139th Psalm, I praise you because I am fearfully and wonderfully made. Your works are wonderful. I know that full well. I hope you've enjoyed tonight's talk, and we look forward to seeing you next time. Thank you.